I do have time. Okay. I don't know if I'll be able to get into the research, but what I kind of want to do, and I kind of stitched this lecture together, but I want to give you an idea of like, what are the processes for me as, when I get a gene list? How do I go find this biology? And I think the easiest way for me to describe it is to kind of set it up as a crime investigation, right? So you have a crime that, say, in my example, the bank is robbed, right? The bank's in the middle. And all of these individuals were around the bank or near the bank when it was robbed. These are actually my genes, this in my analogy, right? And you are the investigator. That as soon as the bank was robbed, the alarm went off, there was cops that were really close. They basically secured the scene pretty much right after the bank was robbed, right? Or very shortly after. And you are the investigator. You got to figure out who's guilty and who isn't. And at your disposal also is the database for Scotland Yard, right? Because we know things about, not only do we know, do genes have a history, but so do people, right? Where would you expect most of the guilty individuals to be in this example? Near the bank, yes. Totally, right? That as soon as the bank was robbed, they're probably trying to get away. If they came in quickly, most of the people are around. Maybe this guy is really, really fast and ran farther than everybody else, right? And with statistics, this is what we do, is that basically it's us basically drawing a perimeter around these banks, right? I can assign a p-value. I can say, I could do a p-value of 0.01, and I can use any test I want. I can t-test, correlation. But basically what we're saying is that most people around the bank are probably guilty, right? And I can draw this line anywhere I want. I can go farther, right? You can see the advantages that if I go farther or I'm less stringent on my p-value, I'm getting more guilty individuals, but I'm also getting more false positives. And that's a problem with bioinformatics, is that there are a lot of false positives. How do we get rid of these individuals that were maybe just walking by the bank, on their lunch break, and they're not guilty in the crime? And that happens with these gene lists. But what most people do is they draw it very, very close. Have you guys gone over false discovery rates? Bonferroni, Benjamin Hoffmeyer, a bunch of other old white men? No? Okay. So p-values work at a gene-to-gene basis, right? Probability, when you do a p-value, that's a probability of just that test. The problem with bioinformatics is we're running, say, 55,000 t-tests. That error combines, it accumulates, and so that you have to correct for all these tests. And so what most bioinformaticians do is they do a false discovery rate, and that applies to lists, saying out of these 10 individuals around the bank, I'm going to do an FDR of 10%, so that only 10% of them should be just your random bystander. Okay? And again, we can kind of use crowds for this too, right? That there's not just more than one bank. That there's also, just like there's more than one crowd. That if, say I had this individual and he was walking by 3 banks that just happened to be robbed at the moment, does the probability that he was guilty go up? Totally. And we do this a lot. That if I put it, I'll analyze 7 different lung cancer data sets to just get me one gene list. This works really, really well. Okay. So we can draw fences around. We can really concentrate on the people that were close to the bank. What other information? Or again, you're the investigator. What other information about these individuals will tell you? And you get to interview them. You can do whatever you want. What other information would help you determine who's guilty and who isn't? Remember, Scotland Yard.
And the means to do it?
The means?
Yeah.
How would they have the means?
Like,
Inclination, maybe, right?
yeah.
What would tell you if they maybe were inclined to rob the bank?
They were under financial stress.
I think you're getting a little too specific, but I like where you're going. Anybody else?
Their history?
Yes! Correct, right? Criminal history. If he's been guilty of robbing a bank before, these individuals have been guilty of robbing a bank before, and they just happen to be walking around the bank, you know, are the odds that they maybe committed this robbery go up? Right. Does it necessarily mean that they committed this crime? No, right? And just because somebody here doesn't have a criminal history, it also doesn't mean that they couldn't possibly be guilty of this crime. But we do this in bioinformatics, is that these genes that are associated with cancer are usually, with lung cancer, are usually associated with other cancers. You're looking for, the usual suspects in a way. This is a graph. So all these boxes are representative of the size or the number of genes. This was a gene list I looked at that were differentially expressed in invasive bladder cancer. And they're kind of grouped by biological process. If it's orange, that means we predict that process to be more active. If it's blue, we predict it to be more inhibited. And as you can see, there's lots and lots of cancer. Things associated with cancer, blood supply, right? These are all going up. What are the only things that are really going down in this? Cell death, right? And that makes sense with cancer. That's why cancer survives, is it doesn't die. And these genes could probably get you to some of those genes. It doesn't mean that they're potentially operating your system, but it's a good place to start looking. Okay? Okay. So we talked about criminal history. What other information would help you determine who's guilty or who isn't?
Appearance.
Racist. No. Sorta. Well, let's talk about, stop thinking that these is just individuals by themselves, Think about something else. What else would help you? Again, stay with that kind of criminal history thing. Know somebody? Yeah, that's coming. What if they're in the same gang, right? What if they're in the same mafia group or something like that, right? That say you had 9 people in this particular gang and 7 of them just happened to be walking by the bank when it was robbed. Did the odds that those 7 committed the crime go up? I think so. What? I think so. They're just hanging out. And that's what we do. So this would be an example. So all these individuals in the blue are known to be in a gang. They were by the bank. They gather more interest in us. And this is actually what pathways are, right? We've established these prearranged, interactions between these things, that these things are all working together usually, and they all show up here. The odds that it's probably involved in our system go up, right? And that's the thing. These gangs can be huge. They can be small and powerful. They can be doing terrible things in your cell, right? But the thing is that these gangs usually share gang members. So which gang do you go after? Number one. Number two is, remember this is Scotland Yard's version of the gang. And your ability to identify a gang is, based on your intel. And a lot of this intel we have on these pathways is pretty old. So, it can give you a good idea, but it might not necessarily be exact. And this just kind of gives you an example. These are the pathways that came out of aggressive versus non-aggressive lymphoma, the genes that were different. And you can see anything with a line shares genes that are similar to those pathways. Some of these, share 8, 9, 7. Which pathway do you choose? But what you can see here is that you've kind of got an immunological pathways, these are kind of the alarm for, these are specific signaling immunological pathways. And then everything over here, even though they have connections, they're all their different pathways, they're all kind of immunological. Basically what this is telling me is that there's a lot of immunological stuff involved in aggressive lymphomas. And we see that everyone here has probably had cancer before, right? Everybody looked up like, what? Yeah, it's your immune system actually takes care of it before it becomes a problem. And what happens in these aggressive tumors, you have to somehow suppress, you have to hide from your immune system. And you see that a lot in these tumors. I see a lot of immunological stuff. And that's kind of a hot area of cancer work now. Okay, networks. So we've got criminal history. We've got known gangs. What else? And it was kind of going back to what you said. Or I think it was you. What else? What other information would help, right? We have criminal history, criminal associations. What about just any association, right? So say, let me give you an example. That all those individuals in the yellow, right? Say 3 of them have a criminal history, 2 don't. But here's the thing, is that say these 3, is that 3? 4. These 4 work at the same, I don't know, tire shop. And this person is the sister of this guy. There's a relationship there. And that's kind of like that network that I showed you, right? These are new gangs that we didn't know about, right? Finding the new gang, put together to do a particular purpose that isn't known to law enforcement. Well, actually, this is kind of a bad example because they had a, who is it, Mr. Pink? That was the informant? Okay. All right. So how does this work in, and that's kind of what I think is the most important thing in bioinformatics is not validating what we already know, but going that step beyond. What are these different relationships? And that's where we come to these networks. So as an example, I made a 3-dimensional network of bladder tumors, right? So at the time, it was the most extensive sequenced bladder tumors, or the number of bladder tumors in the world. So it was about 110. So we had all the genes that were mutated in each tumor, number 1. And then we basically had that for all tumors. And there's more than one gene that's mutated in a different tumor. How would you make a network that would actually describe that? Any ideas? Actually, since I'm running out of time, I'm just going to tell you. So mutation rate, the more the times you see that gene mutated, what I said was, okay, let's make that bigger, right? And this is just network stuff, right? So these are my nodes. So let's make those things bigger. The more time I see P53 mutated, the bigger that node's going to get. And here's the thing that most people don't know or don't do is that if I have a gene that's mutated and this other gene, this different gene, seems to be mutated in the same tumors most of the time, that's important information, right? Maybe they're working together again. So here's what I did is, the more frequently 2 genes were mutated in the same tumor, I made that distance shorter in my network and the lines thicker, right? Yeah, I love that little thing. And this is what we did. We published on this. That you can see here, it's almost like a 3-dimensional view of bladder cancer. The farther you go in, so anything red, the more red it is, the closer into that core it is. The more green, the more peripheral it is. And you can see here that the things that are mutated the most and more central in these networks are, guess what, P53, but there's this other thing, this KDM6A, and that's important in chromosomal structure, right? If you're going to if you want to cause cancer, you got to make big, big changes. And basically, it's kind of like taking a skyscraper and knocking down the supports to it. That's what this cancer is doing. It's just a cool way to kind of visualize this. And we actually put this on GitHub. So if you wanted to take a look at this, you could go to GitHub and just play around with the network. I think this is where the future of all this stuff lies. God, I'm running out of time. Okay, Upstream Regulators.